Hi, welcome to this next video tutorial in your general biology course. This session is entitled Cell Structure and Function Part 2. So in Part 1 we looked at the discovery of cells and the cell theory, and we looked at the structural components of prokaryotic and eukaryotic cells, including a lot of organelles. What we didn't get to talk about much were membranes specifically, so that's going to be our focus in this session. So let's take a look at our learning objectives. First thing we'll want to do is look at membrane structure. How are membranes actually put together? And then the main role of membranes is to regulate what moves in and out of a cell or a cell compartment, so we'll need to talk about how that happens. Simple and facilitated diffusion are two common ways that substances move in and out of cells. We'll also want to talk about osmosis, which is specifically how water is going to move in and out of cells. And then finally, we'll want to look at active and bulk transport, which are just a couple of other means by which things move in and out of cells, but which require energy. So let's go ahead and get started. Let's look at this video. I think we've seen this before. This is a common paramecium, and I chose this video just because it's so enlarged, and you can see the outer boundary of the cell quite well. Well, that outer boundary contains the membrane, the plasma membrane of the cell. By plasma membrane, we mean the membrane that defines the cytoplasmic boundary. But for the longest time, people did not understand what the structure of a membrane was. And so that's what we want to look at. And first we want to figure out who figured out the structure. Who figured out exactly what membranes are made of? Well, the credit for that goes to these two scientists, Seymour Singer and Garth Nicholson. And in the 1970s, they proposed a model for the structure of membranes, and it's called the fluid mosaic model. So let's take a look at this model that they proposed. Here is a diagram of the fluid mosaic model. On the bottom of this picture, we are on the interior of the cell, where you can still see all of the cytoskeleton microfilaments tying things together. And then outside the cell, where the area is shaded blue, that's the extracellular matrix that we've talked about. And then, of course, there's the membrane, the plasma membrane that separates the two. And we could label a lot of things in this membrane, and we'll need to talk about all of these parts. But this is the fluid mosaic model of membrane structure that Singer and Nicholson proposed. Let's look at a little bit simpler diagram and see if we can explain what's going on here. So fluid mosaic model, the first thing they said is that the membrane primarily consists of phospholipids arranged in a bilayer. So in this picture you can see all those phospholipids, and we've talked about them a little bit before. Phospholipids are a type of fat where you have the glycerol with two fatty acids, and then in place of the third one we were putting a phosphate group. And so those phospholipids are arranged in a bilayer, and you can see why they use the word mosaic, because it literally takes a gazillion of these things all set down next to each other to form the membrane. It's a little bit like a mosaic made of glass, where you have lots of different colored pieces of glass that you put next to each other to create a larger picture. So it's primarily a mosaic made of phospholipids, but it contains other things as well. So it also contains proteins, carbohydrates, and sterols. So you can see the little carbohydrate chains dangling up there, those little green sugar chains, the large purple colored proteins, and then those sterols wedged into the middle of the membrane, cholesterol being the one that we see here because that's what we find in animal cell membranes. So we need to talk about the roles of all this. But the membrane at large has the job of regulating what enters and exits a cell, or for that matter, a cellular compartment. I keep referring to the plasma membrane. That's the one that defines the boundary of the cell. But the mitochondria has a membrane, lysosomes have a membrane, the endoplasmic reticulum is lined by membrane. And so membranes regulate what enter and exit these compartments as well. Now let's think about the fluid nature of this arrangement. So here is just the phospholipid bilayer component. And again, I know we talked about this a little bit earlier, that phospholipids are both hydrophobic and hydrophilic. The fatty acid tails are hydrophobic. They do not mix with water. It's pretty much just that hydrocarbon backbone there. But the hydrophilic heads do mix with water. That's where the phosphate group is located of the phospholipid. And phosphate groups, as you recall, bring a negative charge. And anytime you have charge, you are going to be hydrophilic. So the phospholipids just spontaneously spring into this bilayer. In fact, if I had a handful of phospholipids in my fist and I stick my hand under water and just let them go, the laws of physics would take over and they would just spontaneously form a bilayer like this so that the hydrophilic heads can point towards water and the hydrophobic tails can get away from the water. So they spontaneously form membranes like this. But the membrane also has a viscosity, which is another term for fluidity, and that viscosity is similar to olive oil. So it has a texture to it. And so by fluid, we also mean that it is a bit dynamic. The phospholipids, first of all, are spinning. I know in the picture they're just kind of sitting there, but you need to imagine them spinning in place. And not only are they spinning, they can move laterally, that is horizontally. So think of it as a crowd, like at a party where you got hundreds of people standing at the party, but everybody at this party is spinning. Be an odd party, I guess. But they're also moving around, mingling, going over to bump into somebody else and then move through the crowd somewhere else. So they're spinning and moving laterally. And so it gives the membrane this sense of flow or this viscosity, but it does need to be similar to olive oil. We don't want the membrane to get too stiff. We don't want it to stiffen up like butter, and we don't want it to get too runny or it's just going to fall apart. So that fluidity or that viscosity must be regulated. So let's think about how a cell could regulate the fluidity of its membrane. And this will get into some things we've actually alluded to previously when we talked about fats. So here are a couple of examples of the phospholipid bilayer as we attempt to regulate fluidity. One way to control this is to increase or decrease the saturation content. So remember the difference between a saturated fat and an unsaturated fat. Saturated fats had no double bonds between carbons. That meant the fatty acid tails could be nice and straight and you could pack them very tightly. In fact, that's what you see on the top right. That membrane up at the top has got a lot of saturated fats in it. And then unsaturated fatty acids, recall, have a double bond in the fatty acid tails, which causes the kink. We said because of that kink, they don't pack as tightly, and that's what you see in the top left. So simply by changing the saturation content of the membrane, we can change its viscosity. If we saturate most of the phospholipids, the membrane becomes very viscous, very resistant to flow. It stiffens up. Whereas if we increase the number of unsaturated fatty acids, that pushes the phospholipids apart, creating some gaps and spacing, and the membrane becomes more fluid. Now again, we want it to always be the texture of olive oil, so the cell is simply going to increase or decrease the saturation content as needed to maintain that very consistent viscosity. Now, in addition to messing with the saturation content, the cell could also increase or decrease the amount of cholesterol in the membrane. Initially, when cholesterol is put into the membrane, it stiffens it up. You've probably heard about cholesterol being a problem in our blood circulation because it can solidify and clog arteries. Well, it can do the same thing within a membrane. So initially, you put some cholesterol in there and it'll stiffen the membrane up. But as the membrane, if it were to cool down, which is a scenario where we would expect the membrane to solidify, kind of like putting melted butter in a refrigerator, as it cools down, the cholesterol prevents phospholipids from getting next to each other tightly. So it can kind of act in both directions. So it can reduce membrane fluidity at moderate temperatures, but at low temperatures, it actually hinders the solidification of the membrane, which is a good thing. As mammals, we maintain a constant body temperature, and that helps also maintain a constant fluidity. But imagine animals that are cold-blooded, that are kind of at the mercy of the environment around them. They're going to have to really be vigilant about maintaining the viscosity and fluidity of their membranes. All right, so that's the fluid component, really dealing with the phospholipids. Let's go back to the mosaic component, that it's a bunch of different things all stuck in there. And let's start by looking at those membrane carbohydrates. So first of all, the carbohydrate chains, the little chains of sugars, will need to be anchored to the membrane. So they're often anchored to the proteins themselves. And when you anchor a carbohydrate to a protein, like we see in this picture, then what you now have are called glycoproteins, kind of a hybrid, glyco implying the sugary carbohydrate part, and then, of course, protein. So a glycoprotein is really a hybrid between sugars and proteins. And glycoproteins are usually involved with cell recognition. We talked about this previously, that one of the roles of carbohydrates in living things is to help cells recognize each other. And I think the example we gave back then was the AB, and O blood types. So whether you have type A blood, B blood, AB blood, or O blood depends upon which types of sugars you have dangling on the surface of your cells, either the A sugar, the B sugar, both A and B sugars, or type O actually means you don't have either of those types of carbohydrate chains on your surface. But that's the main role that carbohydrates are playing in the cell membrane. Well, what about the membrane proteins? They're doing the greatest variety of stuff. I like this figure a lot because it shows a lot of the various things that membrane proteins do. There are two cells in this picture. The top right corner, you can sort of make out, a quarter of one cell. And then at the bottom, you see the membrane of another cell. And there's proteins wedged in here doing a variety of functions. Let's look at each of those tasks in just a little bit more detail for a second. So here they are broken down separately. One of the things that membrane proteins do is facilitate transport. So we know that membranes regulate what go in and out of a cell. And sometimes we need proteins wedged into the membrane, folded into the shapes of tubes and tunnels or pumps to help get molecules from one side to the other. So transport is an important role of membrane proteins. There are also enzymes embedded into the membrane. And as we've said, and we'll talk a lot about more later, enzymes catalyze or speed up certain chemical reactions. And some of those catalysts, some of those enzymes happen to be anchored or wedged into the membrane. Membranes also play a role in signal transduction. For example, when a hormone shows up at a cell, it's going to show up outside the cell, and it's going to bind to a signal transducing receptor. So we've got this protein wedged in the membrane. The signal, like a hormone, binds to it, and then the receptor needs to convey that message to the interior of the cell so the cell can respond to it. I think of these types of proteins as almost like baseball gloves sitting there in the membrane, waiting for a specifically shaped baseball to bind to it. And when that happens, they send the message to the interior of the cell so the cell can change its behavior. We call that signal transduction. We'll talk about it quite a bit more later in the semester as well. Another job is cell recognition. We've already alluded to this. I mentioned that glycoproteins have little sugar chains dangling off that serve as flags or markers, but there has to be a way for the neighboring cell to read or interpret that marker. And so that's what you see in this picture is one cell has the marker and then the other cell has a protein that fits it. And it is that binding there that's going to help the cell recognize who the other cell is. Another role is intercellular joining, connections between cells. We talked about this quite a bit in the last video. We had the anchoring junctions and the tight junctions and the gap junctions. The one they're showing here is very much like a tight junction where the protein in one membrane is interacting and binding to a protein in the other. And so we've almost riveted those two membranes together. And then finally, some membrane proteins are serving as hubs or anchoring points for the cytoskeleton, both the interior cytoskeleton, but also the extracellular matrix. So if you notice in this picture, we've got some proteins wedged into the membrane, and some of the collagen fibers are tethered to that on the outside, but some of the microfilaments are tethered on the inside. So it's acting as just a hub for both the extracellular matrix and the interior cytoskeleton. So proteins do an awful lot of things in the membranes of cells. And some of the proteins are located on one side or the other. Some proteins, however, span the membrane. So let's make that distinction. So we're saying that some proteins are located on one side of the membrane. Those are called peripheral proteins. Some of the enzymes might exclusively be on the interior side of the membrane, for example. But many membrane proteins, including the one in this picture, span the entire membrane. They go from one side to the other, in which case we call them integral membrane proteins. Recall our discussion of protein structure, primary, secondary, tertiary, and quaternary. You can tell that the protein in this figure has folded up into a tertiary structure, but the way they've drawn the figure for us, we can even see some of those secondary structures, those alpha helices in the middle of the membrane. There's several helices arranged in a circle there to create the overall folding pattern that gets this in there. And when you have a protein that's spanning the entire membrane, it's got to fold in a way to allow for that. In other words, the amino acids in the center of this protein, or in the middle region, they're near those hydrophobic tails. So we're probably going to need amino acids with hydrophobic side chains to be in this area. Whereas amino acids that are near the hydrophilic heads, those amino acids will also need to be hydrophilic. So thank goodness some of the 20 amino acids are hydrophilic. And so we got to make sure the protein is folded in a way that puts the right amino acids in the right places to allow for this type of event to occur. Now the membrane as a whole, its main job is to regulate what enters and exits the cell. And we would say that the membrane shows selective and differential permeability. So what do we mean by those two terms? Those three terms: selective, differential, and permeable. Let's start with permeability. The word permeable simply means ability to pass through. Something can permeate, it can pass through. Right now I happen to be wearing gas permeable contact lenses. That means they are able to allow carbon dioxide and oxygen and things to permeate or pass through. Makes them much more comfortable to wear. So that's what permeability means, the ability to pass through. But what about selective and differential? Selective means that the membrane is choosy about what can pass through. It's kind of a yes or no question. Some substances can pass through, other substances cannot. So the membrane is going to be selective. What do we mean by differential? Differential means that if you can pass through, some things pass through easily, some not as easily, some pass through quickly, some not as quickly. There are different rates or abilities to get through. So selective is kind of a yes or no, can you get through or not. Differential is more of a, well, how easily do you get through? So let's think about what features of a substance might determine whether it gets through or at what rate it gets through. So small nonpolar, and another name for that is hydrophobic. So small hydrophobic or small nonpolar molecules, they pass through easily. If you're small, then you can fit in between the phospholipids and just kind of sneak between the layers there. But you can't just be small. You also have to be nonpolar or hydrophobic, because the bulk of the membrane is those hydrophobic tails in the center. And so you're going to have to be able to engage those hydrophobic tails by being hydrophobic yourself. So you've got to be small and hydrophobic to get through the membrane easily. If you're a large and or polar or hydrophilic substance, you're going to need a little bit of help. Large molecules are just simply too big to fit between the phospholipids, so they just can't wriggle between there. And even if you're small, if you're polar or hydrophilic, you're not going to be able to get through the fatty acid tails. So if you're large and or hydrophilic, you're going to need some help. Help could come in the way of those proteins. Some proteins can fold up into the shapes of tubes and tunnels to help substances pass through. So as things move through the membrane, we would call that membrane transport, and there are 4 mechanisms by which substances move through the membrane, 4 mechanisms of membrane transport. And we're going to need to talk about all 4 of those. The 4 mechanisms of transport are simple diffusion, facilitated diffusion, active transport, and bulk transport. So let's work through these 4 things. But before we can go through any of those, we're going to need to talk about something called a gradient. So that's where we need to go first. So what do we mean by a gradient? A gradient is an unequal distribution of something. You've got more of something in one area than you have in another area. So if you have an unequal distribution of something, that's a gradient. It reminds me, for example, when I'm driving through the mountains, I'll sometimes see a sign that looks like this. Sometimes there'll even be a percentage under there, like 6%. But what this sign is telling me is that there is an unequal distribution of height. And I better get ready to use my brakes because things like to go down gradients. Things like to go from where there's a lot of height to where there's less height. So an unequal distribution of something. It could be height, but we could also have an unequal distribution of heat or energy. That would be a thermal gradient. We could have a pressure gradient, areas of high pressure and low pressure. That's what allows you to fill your car tires with air at the gas station. You could have an electrical gradient. That's what a battery is. Notice on the battery how they've labeled the plus end and the minus end. It's almost like they've put all of the negative charges, all of the electrons down there at the bottom, and at the top there aren't really any electrons, so that takes on the opposite charge, or positive. So we've got this unequal distribution. A battery is a gradient, and just like the truck going down the hill, things like to move down their gradient. So if all the electrons are at the bottom rather than the top, they would like to get to the top, but there's a barrier in there preventing it. And so the battery can't really do much. But if we were to put a piece of metal between there, now the electrons can flow down their gradient, and as they travel, we might stick something in their way like a light bulb, because as they travel through this, we can harness the energy of this gradient and get some work done. We can get this thing to light up. So batteries are a type of gradient that's storing energy, and we can take advantage of the gradient when we let the gradient undo itself, or let things flow down the gradient and get energy out of it. So a battery is interesting, but the one that's going to matter the most to us in this course probably is a concentration gradient, which is an unequal distribution of a substance. So let's look at this picture for a second. Here is an unequal distribution of a substance, whatever that red dot represents. Maybe it's sugars, maybe it's salts, maybe it's amino acids, who knows? But that's a substance or a chemical, and clearly from this picture we have an unequal distribution of it. So we have a concentration gradient. But now let's make sure we understand what the word concentration actually means. So when we say concentration, it's actually two numbers or two values. You can express it as if it were one value. You can say, well, the concentration of something is such and such. But that value is derived from two numbers. It is the amount of solute per or divided by the amount of solvent. So we need to think about the definitions here of solute and solvent, because it's a ratio, it's a proportion of solute to solvent. Solutes, like salt or sugars, get dissolved in solvents, like water. So if I had a glass of water, that might be my solvent, and then I could take a teaspoon of sugar or a teaspoon of salt and dump it in there, and the salt would dissolve in the water. So the salt would be the solute, the water would be the solvent. And in my case, water's the solvent. I could have a glass of, I don't know, alcohol or something and stick something in that, in which case the alcohol's the solvent. And whatever I stuck in, it's the solute. So solutes get dissolved into solvents. So in this picture, these little red dots are the solutes, maybe salt ions, and the white space represents the solvent or the water. And you can see we have a concentration gradient, because over here we have a lot more solute to solvent than we have over here. On the right side, there's a greater proportion of solutes to solvent than on the other side. More red dot per white space on the right than there is on the left. So we have a concentration gradient. And again, just like trucks going down a hill, these solutes are going to want to move down their gradient from their high concentration area to their lower concentration area. And if they do this, if they just naturally start going down their gradients and moving from higher concentration to lower concentration, that's called simple diffusion. So that's our first mechanism of transport. We could define it this way. We could say simple diffusion is the net movement of a substance, a solute, from areas of higher solute concentration to areas of lower solute concentration. So in the picture there to the right, you can see that the solute is more concentrated outside the cell than inside, and so it is diffusing across the membrane from higher concentration to lower. But bear in mind that if you're just going to cross the membrane like that, wriggle between the phospholipids, those red dots better be small. They appear to be small. Looks like they could fit in between. But they also need to be hydrophobic or nonpolar so that they can get through there. And by the way, this process of simple diffusion is passive. What that means is there is no energy required to do this. A truck will naturally go down that gradient. You could, as long as the wheels are able to turn, the truck will roll down. You don't even have to have gasoline to pull that off. Now, if you want the truck to go up the hill, that's a whole nother story. But trucks will passively go down gradients, and just like that in simple diffusion, this substance will move down its gradient passively. But let's go back to the definition for a second. I said net movement. What do I mean by net? Net means that when all movements are said and done and taken together, the majority of substances have moved from the higher to the lower. Because interestingly, the solute can move in both directions. I realize the arrows in this figure are all moving in, but it is very possible for those solute substances to move out. So what net means is there'll be more moving in than moving out. So the net change is that stuff has moved into the, at least in this picture, into the cell. If the gradients were reversed, something could easily move out of the cell, but in this picture they're showing the higher concentration outside the cell. So let's go back to this picture again. So we would say, well, here is the higher area concentration. Here is the area of lower concentration. Now, red dots are going to move everywhere. They're going to move right to left. They're going to move left to right, top to bottom, bottom to top. They're moving all over the place, but because of the gradient, the net movement, the majority of the movements are going to be from the higher to the lower. And if we let that play out over time, eventually it will start looking like this. And now the gradient has disappeared. Everything's still kind of moving around, but the gradient's gone. So even all those random motions, we're not going to see the gradient recreate itself. So we've kind of reached equilibrium at this point. Now that was accomplished by moving red dots. But an interesting thing to ask is, can the solvent diffuse? In other words, can the white space, can the water molecules move as well? Would it be possible to move the water from one side of this box over to the other side? And the answer to that is yes. But we use a different word when it is the solvent that is diffusing. When the solvent's moving from its region of higher solvent to its region of lower solvent, we call that osmosis. So osmosis is the diffusion of solvent from areas of higher solvent concentration to areas of lower solvent concentration. So the solvent in cells is always water. That's what the main component of cytoplasm is, and blood, and living things are mostly water to start with. So that's our solvent. So notice those little Mickey Mouse looking blue things. Those are water molecules. And in this case, there apparently are more of them on the outside than the inside. So the net movement of water in this picture is to the inside. And it is also passive because, again, it's following its gradient. It's going from where there's a greater concentration of water or solvent to where there's a lower concentration. So that's still passive. All right, let's look at an example of how this might work. And we need to introduce a couple of terms too. Let's say that I've got this bacteria cell. That's the orange capsule-shaped thing. And the bacteria cell is swimming around in an environment shown in the blue area. Well, look at the concentration of black dots inside and outside the cell. It appears to be the same. So we would say that we have isotonic conditions. Iso means same or equal. Tonic means tension. So the pressure, the tension, the concentration is the same both inside and outside. We have an equal solute concentration inside and outside the cell. So the question would be, when the concentration is the same, which direction is water moving? Which direction is solvent moving, well, it's the same. It's the same for the solute and the solvent. So water moves equally in both directions. Water's going into the cell, it's coming out of the cell, but it's really the same amount going in and out, so there's no net change. And that's fortunate for the cell, because if it was gaining or losing water, it would ultimately be changing the shape of the cell. So that's isotonic. Equal concentrations of solute in and out. Notice the term's referring to solute, but I'm asking you about the water. So the term isotonic means equal solute, but in isotonic conditions, water, solvent, moves the same in both directions. Well, what about this scenario? What if the outside environment has a greater concentration of solute, black dots, and the inside of the cell has the lower concentration of black dots? Well, the inside of the cell would be described as hypotonic. Hypo means less than or beneath or below. So we're saying that the solute concentration, the black dots, is lower or less than or beneath in the cell. And then outside the cell, in the environment out there that the cell's living within, the environment would be considered hypertonic. Hyper means excessive, more, greater than. So hypertonic is where we find the higher solute concentration. But let's ask about water. Bear in mind that the solute and solvent concentrations are always exact reverse of each other. Wherever the black dots are concentrated, that's where the solvent, the blue, is less concentrated. So we need to think about where water is going in this scenario. If the cell is in a hypertonic environment, which is what we see, then the net movement of water is out of the cell, because the water is trying to move out there to dilute that down. If the solute is more concentrated outside the cell, then the solvent is less concentrated outside the cell. And that's why water is moving from the hypotonic region to the hypertonic region. Black dots would be going the other way. If black dots could cross through the cell, they'd be moving into the cell. So solutes and solvents will always be going in opposite directions. That's another way to think about it. Water always moves towards hypertonicity, and solutes are always going in the opposite direction. They're going from where it's hypertonic to where it's hypotonic. And then one more scenario. What if we had something like this? Now the inside of the cell is hypertonic. That's where the higher solute concentration is. And the outside of the cell is the hypotonic area. That's where the lower solute concentration is. So black dots want to move out of the cell, but water, the solvent, wants to move into the cell. So in a hypotonic environment, that's what the cell's swimming around in, the net movement of water is into the cell. So this stuff's really important. I hope I'm explaining it well enough to you guys. The terms hyper and hypo refer to solute. Solute and solvent are always going in opposite directions. So water, in this case, is coming into the cells to try to dilute down that hypertonic solute. Well, in these pictures, they're bacteria cells. Let's think of some other examples that might hit closer to home that we can relate to. So this is an interesting picture of a foot, and it reminds me of my favorite time of life. I had a 4-year period between age 20 and 24 when I could dunk a basketball, and I was playing basketball like 3 hours a day. And the problem was I have fairly weak ankles, and I would constantly sprain my ankle and it would swell up like you see in this picture. So this is a swollen ankle, and so a lot of bodily fluids are accumulating. Most of it's water, but a lot of bodily water fluids have accumulated and created the swelling. And the question is, how do you get the swelling down? Well, I would rely on osmosis. I would go to the drugstore and buy some Epsom salts, little crystals. And I would dump the Epsom salts into a tub of hot water, and I would put my foot into this tub of Epsom salt solution. Epsom salts, by the way, are called magnesium sulfate. It's a type of substance. And it's easy to make a gradient because you don't have much magnesium sulfate, really you don't have any, in your body. So it's easy to create a gradient. So out in my foot is the hypotonic, the low solute, really none. And in the tub, it is hypertonic, the high solute. So the black dots, if they could move into my tissues, would be moving into my foot, but I'm more interested in which direction water is going. Remember, water wants to move towards hypertonicity. So as a result, the water moves out of my foot, and as the water leaves, that's my blood fluid water, as that water exits by osmosis, the foot shrinks down and I've reduced the swelling. So a lot of people use Epsom salts to reduce swelling. Now I want to talk about one more really important example of how osmosis impacts you. And that has to do with your blood. So here are three kind of artificial scenarios. Think of that as three beakers filled with blood plasma. And then let's put some blood cells in each of these scenarios. Now let's think about three scenarios that we could have with regards to solutes and solvents here. One possibility is an isotonic scenario where the environment, the solution there, has the same solute concentration as the solutes inside the cell. But it's also possible we could put that cell into a hypotonic solution, or your blood could be hypotonic relative to the cell. Or we could have a hypertonic scenario where the blood plasma is hypertonic, higher solute concentration relative to the cell. So what would happen in these scenarios? Well, in the isotonic scenario, water would enter the cell and exit the cell in the same amounts. And that's exactly what we want. We want fluids, water to move in and out equally so that the cell itself does not change shape or size or become damaged in any way. So that's the normal scenario there. But let's look at the hypotonic scenario. Well, in this case, water is going to enter the cell predominantly by osmosis, and the cell is going to lyse. It's going to fill up like a water balloon and burst. And obviously that's going to be a serious problem. Well, what about on the far right? What if the blood plasma becomes hypertonic to the cell? Well, in that case, water is going to leave the cell, moving towards hypertonicity, and that's going to cause the cell to shrivel up. Both of these problems on the ends, the hypotonic blood and the hypertonic blood scenario, are bad because our blood cells are rupturing or they're shriveling up like grapes. That's why it's so important that you maintain isotonic conditions with your blood sugar and your electrolytes and your sodium and all these various things in your blood. Again, we want to maintain something called homeostasis. I think I've alluded to that word several times, but this might be the first time we've thrown it up on the screen. Homeostasis is this tendency for conditions to remain stationary or static or unchanging. So we want the conditions in our blood to remain very isotonic so we don't run the risk of rupturing blood cells or shriveling up blood cells. In fact, there's some problems that arise if our blood is not isotonic. And the example I'll throw out there is diabetes. Diabetes results when a person's blood sugar levels are not regulated and the concentration of sugar, specifically glucose, in their blood plasma is greater than the concentration of the blood sugar in the cells. And so let's think, what scenario would that be? That would be this one, a hypertonic solution. If the blood sugar, the glucose, the solute gets higher in the surrounding area there, that's hypertonic. And under those conditions, under the conditions of diabetes, blood cells are shriveling up and losing water, and there's all kinds of problems that result from that. The person's blood is no longer isotonic with regards to blood sugar, specifically glucose. As a result, people who are diabetic constantly have to be monitoring their glucose levels and taking insulin as needed to try to maintain that isotonic homeostatic scenario. All right, we've said a lot about diffusion and osmosis. Let's move on to some of these other mechanisms of transport. And the next one we want to talk about is facilitated diffusion. we said that a substance can diffuse simply across the membrane if it's small and nonpolar, if it's small and hydrophobic. But what if it's not? Well, facilitated diffusion is the movement of a substance from areas of higher concentration to areas of lower concentration with the help of a transport protein. So notice here these little green things apparently are charged. They're positively charged. So even though they're small, they're not going to be able to wriggle between the phospholipids. So they're going to need some help. So we've got a protein folded into the shape of a tube or a tunnel, and these little green molecules can simply go from higher concentration to lower concentration, moving through the tunnel. So they're still following their gradient. They just needed a little help to get across. And a good example of some common things that move in and out of cells by facilitated diffusion are glucose and amino acids. They're small, they could fit between phospholipids, but they have so many polar covalent bonds that they just can't interact with the fatty acid hydrophobic tails. So glucose and amino acids, whether they're coming in a cell or out of a cell, are going to need the help of this transport protein. Bear in mind again, though, they're still following the gradient higher to lower, so this is still passive. You still don't need any energy. If the truck is going down the hill and there's a wall that would stop it, all we got to do is cut a hole in the wall, cut a tunnel, and the truck can still passively move down that hill as long as it had that opening to go through. So facilitated diffusion is still passive. Now, so simple and facilitated are passive, but that's a good way to transition now to talking about active transport, which is the opposite of this. So active transport is the movement of a substance from areas of lower concentration to areas of higher concentration. We're now going against the gradient. This is like the truck going up the hill now. So we're going from the lower concentration to the higher, the exact opposite of what the gradient wants to move. And as a result, this requires energy. It takes gasoline, after all, to make the truck go up. But we can't just have gas. We also need the truck engine. We need the motor to move it up there. The motor is fueled by the gasoline. In cells, we need energy. It often comes in the form of ATP. We've said ATP is that immediate energy source. But then we also need a protein to serve as the pump. We need a protein to be the engine. Now, interestingly, active transport is often used to create gradients, to create unequal distributions of things. And the example that I'm showing you in the picture is one called the proton pump. That refers to that protein wedged into the membrane, that purple thing. That's the protein. And it is a pump. It can pump protons out of the cell. Protons are those positively charged gray particles. Now, one thing to bear in mind, some people call this the hydrogen ion pump because protons and hydrogen ions are exactly the same thing. Let's think about that for a second. A hydrogen atom is a proton, one proton in the nucleus, with an electron orbiting around it. And if the hydrogen atom were to lose its electron, all it would have left is the positively charged proton of the nucleus. And so a loose proton is the same thing as a hydrogen ion. So this proton pump is forcing out protons, or call them hydrogen ions if you will. And so that's going to create an unequal distribution of charge. Because as more positive hydrogen ions accumulate outside the cell, that area becomes more positive relative to the inside. And it's almost as if we've turned the membrane into a battery. The outside of the membrane is positively charged, the inside is the opposite or negatively charged. So we've used active transport to create a gradient that can store energy in the same way that the battery can store energy. But it's also a pH pump. Because remember, pH is a measure of hydrogen ion concentrations. So as we force these hydrogen ions out, not only do we create the electrical gradient, we also are creating a pH gradient because we now have a lower pH out here. Remember, the pH scale, the lower the pH, the more acidic, but that also means the greater the hydrogen ion concentrations. Wherever you have more hydrogen ions, that's where you have more acidity and lower pH values. So this proton pump is creating a hydrogen ion concentration gradient, it's creating an electrical gradient, and it's even creating a pH gradient all in one. And we won't get into it here, but life has harnessed the proton pump for all kinds of purposes. You have proton pumps in the cells that line your stomach. In fact, that's how you generate your stomach acid. You pump these hydrogen ions across the membrane into the lumen area of your stomach. In fact, some people's proton pumps in their stomach work too well and they generate too much acidity, and then we got to give you a drug. We might give you a proton pump inhibitor like Prilosec or Zantac or Prevacid, Nexium. These are all proton pump inhibitors. They shut down this mechanism of active transport, and so you don't generate as much acid in your stomach. All right, we'll encounter other examples of active transport through the semester, but let's move on to our last example of membrane transport, and that is bulk transport. So bulk transport refers to the movement of large or numerous materials across the membrane. And there are two ways you might be moving into the cell or out of the cell in bulk. So we could have endocytosis. That's endo means within, cyto cell, cis process. So the process of moving things within the cell. So if we're moving something large or simply a lot of things at once into the cell, that's endocytosis. And there's two versions of it. This first version is specifically called phagocytosis. This is a type of endocytosis. Phago means eating, so this is the process of cell eating. And what you see in this picture is a cell engulfing a very large food particle. That food particle would not be able to fit between the phospholipids. It's even too large to go through a protein tunnel. Maybe it's an entire bacteria cell that's being engulfed. You've got white blood cells that engulf bacteria cells this way, so this is cell eating or phagocytosis. The other type of endocytosis is called pinocytosis. Pino, the root word there, means drinking, so this is the process of cell drinking. So rather than engulfing a large bulky item, what the cell's doing on the right here in pinocytosis is simply gulping in some of the surrounding extracellular fluid. So you see those little green dots and purple triangles, those represent solutes, maybe energy molecules or something that's just circulating around outside the cell. And for whatever reason, the cell can't bring them in. Maybe the gradient's not in its favor, maybe it doesn't have transport proteins to help. So it's just going to gulp them in by forming a little vesicle there, kind of pinching in and just sort of sucking in a gulp of that surrounding fluid. This, in fact, is how you bring a lot of vitamins into your cells. You don't have ways of transporting them directly, so you bring them in bulk by this bulk transport. So phagocytosis and pinocytosis are both examples of endocytosis because you're bringing things within the cell. Well, sometimes the cell needs to move things out in bulk or something bulky out. And so that would be exocytosis, because exo means external or outside. So exocytosis is the movement of material out of the cell in this bulk method. We've already talked a little bit about exocytosis, although we didn't use that term. I used the word secretion at the time. But secretion is a type of exocytosis. And if you recall, we saw it when we were discussing proteins getting secreted out of the cell. Remember, they were made at ribosomes at the rough ER. They were put in transport vesicles, came over, melted into the Golgi. The Golgi processed it and then put it into a secretory vesicle and secreted it out. And so that's actually exocytosis going on right there. So here's our exocytosis event taking place. Now let's look at another diagram that shows an interesting feature of this, because here we can literally see the phospholipid bilayer of the membrane involved. And what I want you to notice is that in the top figure, we're watching endocytosis. More specifically, you could probably say now that that's pinocytosis because you know the difference. So we're kind of pinching in the membrane and then breaking that off into a vesicle that's been internalized. So we've brought some of the green dots in. But notice at the same time we lost part of the membrane material because some of that plasma membrane got pinched away and became an interior transport vesicle. So you actually lose cell membrane when you do endocytosis. Now look at exocytosis in the bottom figure. We've got a vesicle full of something, maybe insulin, protein, or something that needs to be secreted. And as that vesicle moves towards the plasma membrane, it just sort of melts into it and spills its contents out. But notice that the interior vesicle has now melted into and become part of the plasma membrane. So you're actually adding membrane when you do exocytosis. Cells have to regulate how much membrane they have, so sometimes they'll do endo and exocytosis with nothing inside. They're not actually taking anything in or releasing anything. They just pull in vesicles or release vesicles simply to add or remove membrane from their plasma membrane as needed. So interesting stuff going on. Bulk transport, by the way, does involve energy, because you've got to move the cytoskeleton and stuff around to get these vesicles to move and pinch and break off. So actually energy is involved in both endo and exocytosis. All right, so we've talked quite a bit about membranes now, and that concludes our part 1 and 2 on cell structure and function. But we've got plenty more to do, so we'll get into some other interesting features of life as we move into our next video.